Okay, so we're going to start by going over what I think are some of the most exciting developments in the field of single-cell and spatial genomics over the past year. It's an incredibly fast-paced field in both experimental and computational methods development. I have 10 areas to highlight today, 5 computational methods and 5 experimental methods. And the goal is not to go into detail in any one technique or technology, but I hope to really summarize the intuition behind each method, why I'm excited about it, and maybe convince you to give it a try in your own lab. So everything on this list is something that we think that you should be aware of, even and especially if it's in preprint form. We've made a particular effort to prioritize highlighting preprints because many of you may not have seen them yet, but they could be valuable for your work. And if you find these methods intriguing, we hope you'll check them out and read the full paper. And also, we'll make a video of this talk available after the workshop, so you don't need to take notes. And with that, let's go ahead and get started. All right, so I'm going to introduce our first highlight, which is a couple of new technologies that are DIY. Now, if you're doing single-cell RNA-seq regularly, we've all been kind of a bit spoiled by technology from companies like 10x Genomics. We don't really need to be DIY anymore with single-cell sequencing. You can buy a kit, you can push a button, you can get data. It's becoming easy. But it's not cheap, especially for large-scale experiments. And this year, there are at least 2 technologies that offer scalable approaches for large-scale single-cell RNA sequencing. And in both cases, they're instrument-free, and it's possible to run them yourself, which is very cost-effective. So the first is the EasySky technology from Jun Kao's lab. This is a combinatorial indexing technology. It's similar to SplitSeq, which has been commercialized by Parse Biosciences. There's no instrument. It works by performing 3 rounds of split pool barcoding in 96 well plates. And to make this easy, as the name implies, Jun's group did more than 350 optimization experiments. They use only commercially available reagents, and the goal was to maximize the improvement in cost and data quality. And the result of all of these optimizations is now that you can generate a library for more than 1 million cells in just 2 to 3 days, and it costs only about $800 in reagents. So this is really orders of magnitude savings over 10x Genomics. So we think this is a major advance. In their preprint, Jun analyzes millions of cells from the entire mouse brain in both wild-type and Alzheimer's disease models. There's exquisite power to detect rare cell types in both health and disease. You can read the preprint if interested, but I'll say that we're running these experiments in my own lab, and we're particularly excited as they can be used with both cells and nuclei, and they're compatible with high-throughput perturbation screens. Now, another method called PipSeq came out of Ian Clark and Adam Abate's group, and what PipSeq does is it partitions cells into droplets along with a barcoded bead. So that sounds similar to DropSeq or the 10x Chromium system, but when 10x generates droplets, they use a custom microfluidic controller, custom chips, custom reagents, all of which they will provide to you in exchange for money. But PipSeq just adds cells, beads, and oil to a test tube and vortexes it all together. And if you optimize this properly, you get beautiful, even droplets, which are loaded with cells and beads just by using a vortexer. And if you do this in a 50 ml Falcon tube, you can encapsulate and barcode 1 million cells in a single experiment in just a few minutes of vortexing. So this is a new way to do droplet-based barcoding. And the data is not quite as sensitive as 10x, but the technologies integrate very well together. In this case, the authors profiled samples from human breast tissue using either 10x Genomics or PipSeq. You get very concordant clustering and basically identical gene expression markers for either technology, which is very nice to see, especially when applying a technology to primary human cells. Now, PipSeq is also compatible with PerturbSeq and CiteSeq, and again, improves on cost and scalability. So it's exciting to see these DIY approaches. You can run them in your own lab, and either now or eventually as commercial kits. And it does suggest that there will continue to be competition and price drops in the single-cell RNA-seq market. It also suggests that it will continue to be easier and easier to generate large data sets. And that leads us to our next computational highlight. Peter Karchenko noticed recently that the size of single-cell RNA-seq data sets is growing quite quickly, in fact, even faster than Moore's law. And it's becoming routine to generate data sets that exceed 1 million cells. So this is exciting, but it also creates computational challenges. Even for things like clustering, we need bigger and bigger computers with more and more RAM. But it also becomes difficult just to explore our data, to understand it, to interpret it, and even to share it with someone else. And I think this year in particular, the computational community has started to propose a path forward for very large-scale analysis, and in particular, in thinking about ways to compress single-cell RNA-seq data sets. So one approach which we're particularly excited about was first proposed by Bonnie Berger's group and uses a technique known as sketching. So the idea is you have 1 million cells, but you can compress the data set by just selecting a subset of cells, maybe 50,000. Now, you don't want to select a random subset because then you lose all the rare populations. So what you want to do is select a subset of cells that maintains the biological diversity of the data set. And that's what sketching algorithms allow you to do. And once you have that sketch of 50,000 cells, you can cluster and analyze that and then extend the results to the full data set. So it's a really creative idea, and we implement this in our Seurat v5 manuscript and software package that we released last week. We sketch cells by calculating a leverage score. It's a computationally efficient technique for outlier detection. So we can make sure that when we do our downsampling, we prioritize rare outlier populations. Those are the rare cells that we want. And we're willing to ignore maybe a few CD14 monocytes, for example, in a PBMC sample, as they're quite common. And if we use this sketch-based workflow, now we can start to analyze massive single-cell RNA-seq data sets. We can analyze a data set of 1.3 million neurons from 10x Genomics in an hour. We can integrate and analyze a data set of millions of PBMC from Parse Biosciences efficiently. And you can run these analyses on laptops because they are not memory intensive. And really, this ability to sketch or compress data sets is key to this. And Yuhan Hao and Gezi Malla will be talking about some of these techniques later today. And by the way, we don't have to stop at 1 million cells. This is a data set of 12 million cells from the mouse embryo. It's an extraordinary data set, it's the largest we've ever seen from Jay Shinduri and Cole Trotnell. And we were able to process and analyze it in under 6 hours, all in R, getting results that are extremely concordant with the author's original analysis. And it's a good thing that we could process this quickly because this data set was only released yesterday. These are plots that came in from this analysis on the same day the paper was released. So credit to Yuhan for putting this together, and I think it exemplifies how sketch-based workflows can really accelerate large-scale data set analysis. And if you're interested in this, you can check out Seurat v5, which we released last week. Now, Yuhan and Gezi will also be talking about a second way to compress data sets, which is via aggregation. And this is an idea that was first proposed by Amos Tanai's group. And the idea is, if you have 100 single cells that are very similar to each other, why not just pool them all together? And they propose an algorithm to do that, creating something called metacells. And there are a couple of advantages to this approach. The first is that you reduce the number of points in your data set substantially. And the second is that since you're pooling information across similar cells, you really reduce sparsity. So it's a really good idea, although the algorithm does have to make sure not to pool together cells that happen to be in different states. Now, Donna Pear's group built upon this idea with their C-cells method, which was just published, and it has a similar goal. But they apply it also quite a bit to attack and multi-ohm data, which can be even sparser than single-cell RNA-seq. And they show that C-cells not only compresses the data set, but makes it easier to identify relationships across modalities. And we encourage you to check out this work as well. All right, we'll move now to a spatial technology for our next highlight. It's a technology that was just published a couple of days ago called slide tags. But first, I want to introduce two complementary existing methods for spatial analysis. One is spot-based, methods like SlideSeq or 10x Visium. And what you know, of course, is that the way those work is you transfer RNA from the tissue onto a slide where it hybridizes onto an oligo that contains a spatial barcode. So it's transcriptome-wide data because you then sequence that transferred RNA, but it's not really single cell. And then you also have methods like MERFISH, which directly image RNA using probes. So it's real single-cell resolution, but you only get a subset of genes. And so you can either get single cell or transcriptome-wide data. What we really want is to get both. And Cole Trotnell's group proposed a way to do this a couple of years ago. They call it SkySpace. And I won't go through it today, but along with a method from Jimmy E's group, these were some of the first techniques to propose a new type of workflow for spatial analysis. And that's what Fei Chen and Evan McCosco's group at the Broad introduced a couple of days ago, and it's called slide tags. And it builds off their SlideSeq method, which has spatially barcoded oligos on a slide at 10 micron resolution. And in slide tags, those barcodes are photocleavable, so they can diffuse into a tissue section. So instead of transferring RNA from the tissue down onto the slide, you transfer the spatial barcodes from the slides up into the cells. And once you have done this, the cells are naturally barcoded with their spatial location. And then you just associate the tissue. You can extract nuclei and just run single-cell profiling like 10x. And when you do the single-cell profiling, you sequence both the cell's transcriptome and that spatial barcode. And so now you can do your standard clustering analysis, but you can also use the barcodes to tell you precisely where each cell is located. So you get single cell, whole transcriptome, and high-resolution spatial data all at once. The only downside is you don't capture every cell, but that will improve with time. So I think this really is an exciting path forward. This type of strategy in particular, it's easy. For the user, there's no imaging, there's no image analysis. For example, there's no segmentation, which can be extremely challenging. You're just leveraging the ease of existing single-cell RNA workflows that we already have, but while maintaining spatial resolution. And this approach is compatible with diverse tissues, is compatible with diverse omics, including the 10x multiome technology, and even with TCR enrichment. So I won't say more about this now because Jackson Weir, one of the inventors of this technology, will present this work this afternoon, and I highly recommend you to stay on to watch that if you can. All right, so we'll keep jumping around and move now to a computational method on how to compare single-cell RNA-seq samples, for example, between healthy donors and diseased tissue. And this can be difficult, especially in cases where it's challenging to discretely cluster the samples. And so a couple of years ago, John Marioni's group introduced Milo. And this tool, along with others in the community, performed differential abundance analysis on small neighborhoods of similar and overlapping cells. So the idea is that if you run Milo, you can identify exactly where in your data set there is an enrichment or a depletion of cell types from one condition, even if it's in a subset of a trajectory. There is no pre-clustering required. And this has become quite a popular tool for comparing data sets generated from different conditions. But it raises the next challenge, which is how we move beyond differential abundance and start to look at differential expression. And so this year, Alsu Misarova from John's group introduced Milo-DE, and it performs differential expression analysis across conditions without clustering. And the specific goal is to identify cases where a gene is differentially expressed across conditions, but only in a subset of cells, maybe even in a subset of a cell type. And Milo-DE helps to identify specifically those neighborhoods. So this is an example in the mouse embryo. Also compared wild-type cells and Tal1 knockout cells. Now, one major difference in these cell populations, of course, is that there's an entire population that's missing in the Tal1 knockout cells. So that you could discover, for example, with Milo. But also found another difference using Milo-DE, even in a population that's shared between the two genotypes. She found that a subset of genes that are involved in heart development, like TNNI1 or TNNT2, they were differentially expressed. They were upregulated in Tal1 cells, knockout cells, but only in a very transient cell state. And this transient cell state doesn't even represent an entire cluster, but just a small subset. And in fact, there's a whole program of genes that behave this way in this transient cell state, and that's exactly what Milo-DE enables you to discover. Now, there are multiple methods that do this, and I'll also flag a new paper from Wolfgang Huber's group. It's a method called Lemur. And it can also identify specific subsets of cells, even in a continuous context, that exhibit differential expression of a given gene across conditions. So it works a little bit differently, but it's quite innovative. It basically uses a continuous embedding space as a regression covariate in a generalized linear model in order to perform differential expression without pre-clustering. So Milo-DE and Lemur both come with GitHub software packages. You can try them out. And we particularly recommend trying them if it's difficult to define discrete clusters in your data set, either because of the presence of continuous cell states or developmental trajectories. Okay, for our next highlight, we're going to jump to a new technology. And in fact, the next highlight was the inspiration for our t-shirts today and for our logo for Single Cell Genomics Day. And it comes from Vijay Sankaran and Peter Van Galen's groups. And it's a method to try and resolve cells into clones just based on single-cell RNA-seq data alone. And it leverages the fact that mutations in the mitochondrial genome are quite frequent and can serve as a clonal barcode. Now, we've covered this idea extensively in previous years, but in the past, obtaining those mitochondrial mutations was only possible using single-cell ATAC-seq data. But this method, called MASTER, enables you to identify these mutations in single-cell RNA-seq data. And the way that it works is you take amplified cDNA just from a regular single-cell RNA-seq experiment like 10x, and you tile primers across the 15 mitochondrial transcripts, and then you perform targeted sequencing of the resulting amplicons in order to identify mitochondrial mutations. And you get these mutations, you get this information in addition to the cellular transcriptome. And so now what you can do is you can group together cells that have the same set of mitochondrial mutations, and you can group them into clones can make an inference that those cells are clonally related. So this is a sample from human bone marrow, and each one of these rows on a heat map is a set of cells that share the same mitochondrial mutations. And so in this experiment, they detected around 25 different clones in this bone marrow sample. And now what's exciting is that in this example, you can see where different clones that were defined based on their mitochondrial mutations, you can see what cell types they each represent and whether they have similar transcriptomes, which in this case they do. And I want to highlight what's exciting here is that this can be applied to any single-cell RNA-seq sample to learn clonal relationships. You don't need to introduce CRISPR. You don't need to introduce lentiviral barcodes. And you can even apply this to cDNA samples from single-cell RNA-seq experiments that you have already generated, for example, using 10x Genomics, and that are just sitting in your freezer. And we think that's really exciting. And also, because this technology is compatible with single-cell RNA-seq, you can add in additional modalities. So the authors, for example, also add in TCR, or add in the ability to amplify additional transcripts in the somatic genome for genotyping. Now, this is still going to be most valuable in cancer samples, as these samples are here in this manuscript, where there is extensive clonal structure. But I do think the broad applicability here is exciting. And in fact, we'll have a talk from Sydney Schaefer this afternoon that will use this approach to trace cell lineages from metaplasia to cancer. All right, so now we're going to stick on this theme of variants, but we're going to look at it in a very different context. And I think one of the exciting themes, actually from the last few years, is to use single-cell RNA-seq to help interpret GWAS studies, especially to see if we can prioritize cell types of interest that might be influencing disease. So many groups are working on this, and there are a lot of great methods, including methods that we've covered in previous years. But one paper that really struck me this year came from Jesse Eingranz's lab at Stanford, and it struck me because they proposed a systematic approach to attack this problem. So let's say that you start with a set of genome-wide association study variants, in this case, a few hundred variants for coronary artery disease. And your goal is to identify gene programs and pathways that those variants affect and that eventually influence disease risk. And so what Jesse's lab did was they did a massive perturb-seq experiment. They knocked out about 2,000 genes, basically any gene within 500 kb of a GWAS signal. They then used matrix factorization approaches to group the perturbed genes into programs. So for example, they found that multiple regulators, just as an example, EDN1, PDGFB, and CCL2, when you knocked out those regulators, they all affected a similar group of genes related to wound healing. And so now they can try to prioritize particular programs that relate to cardiac disease progression. The top program that they found to be represented in disease progression was a set of genes that were involved in angiogenesis and osmoregulation. That program included 18 different genes that had previously been linked to coronary artery disease. So that was a very strong enrichment and a very striking result. And of course, because they did perturb-seq, they also know exactly what genes regulate these programs. And that led them to focus on a particular complex, in this case, a complex that governs the strength of interactions between cells that form blood vessels. So I'm not going to go into the details of the discoveries that they made here, especially because one of the lead authors, Helen Kang, in Jesse Eingranz's lab, is going to give a talk on this study later today. But I do want to highlight the vision. And the vision is to connect GWAS variants to the genes they affect, to connect those genes to the genetic programs they regulate, and then to connect those programs to the progression of disease. And I think this is a very exciting vision. This type of systematic approach can be run in many different contexts, as long as you have a model cellular system that you can screen in and that you think is relevant to disease biology. All right, so for our next highlight, we're going to stay on the theme of perturb-seq. But this year, groups have been using perturb-seq not just to look at changes in gene expression, but also to look in other phenotypes. For example, to look at changes in post-transcriptional regulation. And actually, one of the first examples of this came last year from Jonathan Weissman's group. And in their genome-wide perturb-seq paper, which we showcased last year, they perturbed thousands of genes. And mostly they looked at expression changes. But they also found cases where after perturbation, gene expression might stay exactly the same. But for example, if you perturb the integrator complex, transcription would fail to terminate at the proper place, and you could get run-on transcription. And you could identify that not just by looking at the total number of reads, but at the location of the reads and where they were mapping. And so what that shows is that you can identify other phenotypes, not just gene expression levels, in perturb-seq experiments. So another beautiful example of this came from June Kao's lab. As I mentioned, their EasySky technology is compatible with perturb-seq, but in their new method, perturb-sky kinetics, they introduced another twist. So here they introduced 4SU into the media. 4SU is a modified nucleotide. It's taken up by cells and incorporated directly into newly synthesized RNA. And it introduces sequence changes during cDNA conversion. So when you sequence a cDNA molecule, you can tell if that molecule represents a new RNA or an old RNA based on whether it includes these mutations. So perturb-sky kinetics also measures the guide RNA so that you can do a perturb-seq experiment. But now you can use these data not just to search for regulators of RNA expression, but also for regulators of RNA kinetics. And the reason you can do that is based on these sort of newly transcribed data sets, you can infer specific synthesis and degradation rates for each gene in each condition. And then you can ask how different perturbations affect those rates. So for example, you can identify perturbations who affect global rates of transcription, many of whom are transcription factors or interact with RNA polymerase. And you can identify perturbations who affect global degradation rates, which include, for example, members of the exosome degradation complex. You can also group RNA binding proteins together and start to reconstruct complexes who work together to regulate the RNA life cycle. So this is very exciting work, and it's out on bioRxiv. My group also became interested in this idea, specifically in the context of cleavage and alternative polyadenylation, or CPA. And so we refer to our work as CPA perturb-seq. Now, a number of groups have realized that based on the location for where reads map in the 3' single-cell RNA-seq data, you can infer which poly A site is being used. So we reasoned that if you did a perturb-seq experiment with the 3' readout, we could see not only how expression levels change, but if we kept track of where the reads were mapping, we could also see how poly A site usage changes. And so a lot of the questions that we typically ask for transcriptional regulation, we thought that we can also now start to ask those questions for post-transcriptional regulation as well. So here's an example locus. You can see that in non-targeting control cells for this gene, we tend to see expression of the long isoform. But when we perturb the canonical poly A regulator NUD-D21, we see a striking shift towards the use of the shorter isoform. It's really clean, it's really beautiful data. We're leveraging one of the major advances of perturb-seq, which is that all of the controls and perturbations are mixed together in the same experiment, so there are no batch effects. And that really helps us to pick up on bona fide differences between conditions. Now, I'm showing you here this example in one gene for one regulator, but of course, we can systematically do this for a large number of regulators. In this experiment, we perturbed more than 40 known regulators of cleavage and polyadenylation. And I won't go into the biological findings here. I certainly encourage you to read the preprint, but also Madeline Kowalski, the lead author, will be talking about this work along with other methods to study post-transcriptional regulation later this afternoon. Now, in this study, we only knocked out a limited number of genes, and we did that because it can get kind of costly to do very big perturb-seq studies. And that leads us really to our next advance, which aims to address a fundamental limitation. When you are doing perturbation screens, either in bulk or alternatively in single cells, the more perturbations you want to characterize, the more samples you need to profile. There is a linear relationship between throughput and cost. And that's a problem if you want to do thousands of perturbations. And so Brian Cleary has been thinking about this for a while, and he's been proposing to exploit an idea from signal processing called compressed sensing and to apply it to genomics. So it's kind of a crazy idea, but I'll try to explain it. Instead of measuring perturbations one at a time, either in single cells or in a single sample, what if you just randomly blend them together? It's a form of sort of randomized compression. So for example, you can implement this for perturb-seq in at least two ways. You can cram multiple cells into a droplet, for example, by overloading the 10x machine. And as a second way, you can infect cells at a high MOI so that each cell receives multiple guide RNAs. So either way, you're going to be blending different perturbations together, so that each time you sequence a droplet, you detect multiple guide RNAs, and the transcriptome you sequence reflects the average effect of multiple perturbations. Okay, but then here's the crazy part. At the end of the experiment, if certain assumptions are met, you can use matrix factorization to reconstruct the effect of each perturbation individually. So basically, you can deconvolute the average profiles you measure and learn the effect of each guide RNA on its own. But the advantage now is that since you were profiling these blended populations or these blended perturbations, you have to profile a lot fewer samples. So cost does not grow linearly with throughput, it grows logarithmically. So it's really a creative idea, and Brian's group implemented this and compared the results with normal perturb-seq using one guide RNA per cell, and then compared those results with the inferences from a compressed sensing experiment. And in the compressed experiment, they had to profile much fewer cells, but after the reconstruction, they were still able to get a reasonable estimation of the original data set. Now, as I mentioned a couple times, there are assumptions here in order for this to work, and it's not a perfect reconstruction either, but this is conceptually and methodologically a really exciting way forward to think about doing massively scalable perturbations. And we're fortunate to have Brian here. He's going to give a talk on this technique later today and how you can use compressed sensing with perturb-seq to do a wide variety of downstream analyses, including integration with GWAS studies. All right, so now I'll jump to a new technology from Jay Shinduri's lab. Jay's lab usually makes this list, and this year is no exception. His lab has been one of the pioneers of massively parallel reporter assays, or MPRA technology. MPRAs are used to assess the function of multiple enhancers in a single experiment. And so it's no surprise that Jay's been thinking about how to extend this workflow to single-cell analysis as well. And they developed a technology called SEQers. And the way it works is you transduce a multicellular system with a library of reporter constructs, each of which measures the activity of a single enhancer. So, this seems kind of straightforward. You do this transduction, each cell will get a subset of these constructs, and then each cell, you sequence, for example, the transcriptome, and then you sequence these reporter constructs to measure the activity of each enhancer. So it seems straightforward, but it's not. And the reason it's not is because there's a technical problem. If you don't detect the activity of an enhancer in a given cell, is that because the enhancer was off, or is it because that enhancer just never got into the cell in the first place? That's a fundamental challenge. And they solve it with a really creative two-part barcoding system, where one barcode just tells you whether the cell received the construct or not, and the other barcode reports on its activity level. They also use the tornado method from Sami Jaffrey's lab, the tornado system, to stabilize that first Pol3 barcode and boost its expression. This is a really good idea that we should be applying more often. But altogether, what this enables you to ask is, for more than 200 enhancers, what cell types are they active in and how active are they? And so that's what they did. They applied their system to embryoid bodies, where you can generate single-cell sequencing data and easily see multiple different lineages, as well as, for example, pluripotent cells. And now you can ask about the activity of each of these 200 enhancers and each of these different cell groups, which is really exciting. So I'll try to walk you through this data. In these embryoid bodies, just from single-cell RNA-seq data, we can see that there are a few different clusters of cells, including pluripotent cells, but also differentiated populations that express the SOX2 gene. From single-cell ATAC-seq data, we can see that there are multiple enhancers that control SOX2 expression that are near the gene. And I've highlighted, or they've highlighted this one here. This enhancer is only accessible in pluripotent cells. But SEQers gives us a way to verify this. We can look at the activity of this specific enhancer, and we can see that this enhancer is not active everywhere where SOX2 is expressed, but instead it's only specifically active in this pluripotent state. So that's a really cool result, especially because we're not just doing it for one enhancer at a time. We're doing this for hundreds, and that's exactly what they did. They can find enhancers that are constitutively active across all cells, enhancers that are cell type specific, and enhancers that explain only a subcomponent of a gene's expression. It really is a multicellular MPRA. So I won't say more about this today. Jay is, of course, one of our keynote speakers. He'll be speaking this afternoon. He may or may not talk about this story, but you can definitely check out the preprint. Just before moving on, I also want to mention that multiple groups have made contributions towards this goal of assessing enhancer activity at single-cell resolution. Craig Lowe's group introduced SE*-seq, which is a similar type of assay, and I won't talk about it more today, but please do check it out if you're interested. All right, and for our last highlight, I want to introduce this by first mentioning a strength and weakness of single-cell RNA-seq. So this is a heat map that we produced from the mouse retina in our very first dropseq experiment in 2015. And I think what this heat map shows is that single-cell RNA-seq is very good at finding which genes change from one cell type to the next, but we also realize that it's bad at figuring out why those genes change. Just because modules of genes are co-expressed, that doesn't necessarily tell us anything about who regulates those genes, or even if those genes are necessarily co-regulated at all. And this is a fundamental problem with RNA data, trying to distinguish correlation from causality. And this year, multiple groups have been trying to address this problem computationally with a twist, and that twist is to use multimodal data. So there are a number of methods that do this. I'm going to start with one of my favorites. This is from Wolf Reich's group, where they generated a large multi-ohm data set from E8.5 in the mouse embryo. And you can see in the single-cell ATAC-seq data that there are particular enhancers that are only accessible in some cell types and are not accessible in others. And now the goal is, can we figure out why? Can we figure out what transcription factors are binding to and are regulating these enhancers? And this is what Wolf's group was working on. And since we have both RNA and ATAC from the multi-ohm experiment, we can start to answer that question. So what they do is they look at the expression of every transcription factor, and they see if it correlates with the accessibility of a particular ATAC-seq peak. Now, usually there's no correlation, but in some cases, they find an excellent correlation between expression and accessibility. And when you see that, you do a secondary check. You ask if the peak contains the correct motif that's supposed to bind the transcription factor. Now, if both of those things are true, you can make an inference that the transcription factor is actually binding in that peak. And they call this method, because it's predicting binding, they're calling it in silico ChIP-seq. And they benchmark this approach with actual ChIP-seq data, and they can see that it works quite well. So in this specific loci, real ChIP-seq data of the transcription factor TAL1 shows two very clear binding sites. And those are located exactly where the in silico method predicted they would be. And this is exciting because, of course, we can now do this for any transcription factor in the genome, and we can do this genome-wide. And so what's fun here is now we can really start to make regulatory statements about a particular transcription factor that binds to a particular enhancer and linking downstream that particular enhancer to a nearby gene. So we're starting to learn causal, fundamental regulatory relationships about which transcription factor are regulating which genes. That's moving far beyond correlation analysis, and it's enabled specifically by multimodal data. All right, so I won't talk about this more today for time. I'll just say that there are a number of methods that have really made pioneering advances here. One is Cell Oracle from Samantha Morris's lab, and Sam will be speaking directly after me, and I imagine she'll talk at least a little bit about that work. Of course, I mentioned in silico ChIP-seq, and then also John Blair from my lab this afternoon will be talking about a number of these methods, including one that he's developed called phospho-seq, which uses intracellular and intranuclear protein profiling combined with additional molecular modalities to reconstruct gene regulatory networks. All right, so that wraps up my overview of recent advances. It's always quite a roller coaster to do this, but it's fun for me to give this talk and to acknowledge and celebrate the really amazing work from this community. Thanks very much for listening. And the last thing that I want to say is today, all of us here are wearing our Single Cell Genomics Day t-shirts. They were designed by Tracy Smith and my group. And they were also inspired by the Meister paper and these sort of mitochondrial clone heat maps, as well as similar plots that Caleb Leroux and Leif Ludwig and others have generated over the years. And I just want to say that this workshop each year is a lot of fun for us. We love to do it. But we really do want to make sure that it has impact and can reach a broad audience. And the best way to do that is for you guys to spread the word on social media. So if you're interested, if you're enjoying the day, please send a tweet. You can use the hashtag Single Cell Genomics Day. We'll monitor the Twitter, and for people who are active, we'll give away and send you for free 10 t-shirts based on people who are tweeting. And we do that every year, and hopefully it's a little bit of fun. All right, so thanks very much to listening for me, and I'm happy to take a couple questions if there's time.
Hi, Rahul. One question we got from the audience was, how close are we to getting a per-variant ontology enrichment, such that we could go from a list of genetic variants to a cell-type resolved biological pathway similar to gene ontology enrichment?
So I don't think we're particularly close, but I think it's a great question. It's a particularly good question for Helen this afternoon, and her lab and Jesse's lab work really exclusively on this question. How can we use single-cell technologies to be able to interpret genetic variants? So I think we're starting to see the first studies that are making impact here. And I'll just make a broader point at a transition that I'm seeing generally in the single-cell field. A lot of the methods that I talked about today, they're not just for clustering or for discovering cell types or doing just kind of traditional single-cell RNA-seq analyses. They're methods that are using single-cell approaches to help us ask big-picture biological questions. And the one you asked just there, How do we go from variant to phenotype? That's a big picture biological question, and it's exciting that we think single cell technology can help us get there.
One last question is, what is the biggest thing researchers designing new single cell experiments should consider?
Okay, so that's a broad question on experimental design, and I think it's an important one. So I think that the answer is something that's becoming increasingly appreciated in the single cell field, or at least one of the answers, which is it's not just about the number of cells you profile, but the number of replicates, the number of donors, the number of samples you profile is essential to being able to maximize your statistical power. So if you, for example, want to be able to profile samples from healthy donors and from diseased tissue, doing an N of 2 or an N of 3 is not going to be very helpful, even if you sequence hundreds of thousands of cells per individual, because you're not appropriately modeling biological variants from one individual to the next. So I think we're seeing a trend in the field moving away from doing a large number of cells on a small number of donors to doing a smaller number of cells, but distributing them over a larger number of samples, which enables you to more accurately characterize and model biological variants. That does, of course, require access to larger numbers of biological samples, and it's great that's becoming the limiting factor now, not just the technology. But I think that's something important to keep in mind for experimental design.